Daniel Garcia. So okay. So Daniel, he's researcher at Carlos III University at the Department of Continuum Mechanics and Structural Analysis. Daniel, in fact, he has developed his career at the University of Carlos III and at the University of Oxford. His research focuses on continuum media formulations combining mechanics and other physics. And now he has just received a starting grant. And well, so Daniel, you can start now.
Okay, so thank you very much for the nice introduction. So let me share the screen. Okay. Okay, so I guess you can see it now, right?
Yes, perfect.
Okay, so good morning, everybody. Well, as you say, I'm much more modeling guy. So what I'm going to present now is some modeling approaches in biomechanics. So we'll present a very recent work on collective cell migration, then move to briefly introduce some brain mechanics. So regarding motivation, when we look at different biomechanical processes, such as cell migration, traumatic brain injury, so these two ones are going the ones I'm going to provide more details, but also in bone healing or cardiac function, especially the last one, we have this electric response strongly coupled to the mechanical pumping of the heart. We realize that mechanics plays a major role. So when we look inside these mechanics, what we realize this is far from being simple, and we have too many different dependencies. So we can have some temperature dependence that modulates the stiffness of the biological tissue. Also, we can see that these biological tissues are not purely elastic, right? So we have some viscous contributions. And regarding these viscous contributions, we have some consequences, such as here, strain rate dependency, so that depending on the rate we apply the mechanical load, we're going to get usually different stiffer response, right? So the higher the strain rate, the higher the stiffer the mechanical response of the system. And this is also intrinsically coupled to this kind of viscous relaxation, so that if we apply a given load at a given rate and then we keep that load, we can observe kind of a stress relaxation with time. And on top of that, we have many other different dependencies on top, just to mention one, so anisotropy in muscles or tendon tissues, where we have some fibers, and we can see that we have different mechanical response if we apply the load in that longitudinal direction or in the transverse. So something common to all these dependencies is that in the end, so this is not just Young's modulus, right? So this is not just some linear viscoelasticity. So we have strong nonlinearities, and this is important not only from the modeling perspective, but also if we are analyzing some experimental characterizations, we can see that, well, not exactly this, but in the true stress to strain, so this stiffness is actually the slope of this curve. So if we are reaching enough large deformation, everything is changing, and we can have even order of magnitudes of difference. So now I would like to introduce some works where we are playing with these mechanics in a kind of modeling framework. So this first one actually is in collaboration with Arrate from Universidad Carlos III of Madrid as well. So we have recently published that in Stream Mechanics Letters, and to be honest, this is quite new topic for me. So really happy to have any comments or feedback from you guys. So when we look at cell migration, from an individual point of view, we know that these individual cells have some protrusions, right? And some inactive stresses with some contraction or expansion. And this is far from being kind of isolated system. We have some interactions with the extracellular matrix, substrate, or even with other cells. So in the end, we have some interaction forces and some internal forces, really. And as soon as we have some links between different cells, all these mechanical forces are going to be transmitted somehow through the whole system. So just to mention two examples of collective cell migration, we have here cancer cell invasion. So this few have to say, I thanks Dennis Beer from Johns Hopkins University to provide here, because we can see how we follow the time event, we can see kind of continuum-like expansion of this kind of collective cell migration. And I think this is quite nice here in this video kindly provided by Xavier Trepal from Barcelona, that we can see how these forces from individual perspective are introduced some forces that are somehow transmitted to the whole event. And something I would like to highlight here is that we can see that we have regions with large deformation. So in this point, we are losing this kind of linear elasticity, and we need to move to other hyperelastic theories. So looking inside this problem, more from a theoretical perspective, we need to identify different governing equations. So I just mentioned here 3, but actually we'll focus on these 2 latter ones. Just mention we can imagine we can have some proliferation dynamics or other chemical diffusion-like processes actually we're incorporating now. So one fact is that we can consider this concentration of contractile actomyosin. So in the end, we're going to have kind of active stress, right, related to that contractile actomyosin, or we're going to have some contraction or expansion. Then we jump to this cell polarization dynamics. So what this is telling us, okay, so your cellular system can change in shape and orient, right? So in the end, all these forces are going to have any kind of directed resultant. And then in the end, we have this mechanical balance that to me is the most important one. This is kind of cocktail where we can put all the different forces together, and then somehow this is going to determine the fate of this collective migration. So having this in mind, we wanted to focus on a very specific problem. And actually we took this experimental data published by Sanger et al. In Science. So basically we have some epithelial monolayers cultured in different substrates. So we can observe here in the left-hand side image that when we have kind of uniform substrate, we can observe kind of collective cell migration in a very symmetric fashion. But once we move to an heterogeneous substrate, we have kind of a stiffness gradient, right? We can observe that this migration is directed towards this stiffer region. So when we look into the literature, most of the experimental works and even some, nice modeling frameworks suggest that this is somehow a kind of stiffness-dependent propulsion force, so that as our system is somehow connected by focal adhesions to the substrate, we can expect higher interaction forces here in that stiffer region than in the softer one, so that we're going to have kind of resultant force directing the migration to that region. But if we look at the, some experimental works in the literature also, we realize that this is not only this propulsion force, but also somehow of a stiffness-dependent polarization. So we can observe that this cellular polarization depends on both the magnitude of the substrate, but also on some stiffness gradients. So at this stage, we wanted actually to provide kind of simple modeling framework to try to test these different hypotheses and determine from a mechanical point of view, what is the implication of each one. So we started with the mechanical balance. So we provide here somehow together these internal forces and external forces. So we will have this internal cellular stress ponderated by the height of the epithelial monolayer. So here sigma is the cellular stress, and T is the traction forces. So here we say, okay, we're going to have this kind of propulsion force that a priori can be dependent on this stiffness of the substrate following these previous approaches. And then the direction of that resultant force is going to be somehow determined by this cellular polarization. And then, of course, as soon as we have any kind of system moving on top of the substrate, we're going to have some friction forces, right, opposing to that with this parameter being this friction coefficient. So once we have defined that, we need to define what is happening inside the cellular stress, right? So basically, we said, okay, let's try to use some hyperelastic theory. So the main idea here is that instead of defining directly the constitutive relation, we're going to define a kind of mechanical potential. So basically, this is the mechanical energy, right, that is stored in the system and we can recover at some point. So using some solid mechanics and thermodynamic principles, we can reach the following consistent relationship between the deformation state of the cellular system, defined by this F, that this is the deformation gradient, and our Cauchy stress tensors, our true stress. And then we can observe here that we have kind of two components, with this first one being a volumetric one, with this P being kind of pressure that we are going to get from boundary conditions and some consistency equations. And then we have here a second term related to the shear, where this mu cell is the stiffness of the cellular system. So basically the shear modulus. So once we have defined that, let me come back here and we say, okay, so these traction forces are going to somehow depend on how this cellular polarization is. And we can expect this is going to somehow change during the whole event. So we decided to provide this kind of governing equation for the cellular polarization. So we are aware there are two other many terms we can include, like dependence with velocity or others. But we decided to provide these two ones to test the different contribution of our hypothesis. So first, this two blue term is saying, okay, so because of this stiffness gradient, we can have some cells that are going to polarize to a stiffer region, right? So then we have this first term that is saying, okay, as all these cells are somehow linked and add each other, we're going to have a kind of diffusion-like process where all these other cells are going to lead to that polarization. So then, even if we keep kind of constant propulsion force, what we're going to have is kind of resultant force, okay, towards this stiffer region that is going to be able to capture this kind of tendency in the migration dynamics. So just at this stage, I'm going to skip all the mathematics. Basically, what we did is implement that in a finite element code for some implicit integration algorithms. So that's say we're solving all the different equations at every single spatial point at the same time, finding for the exact solution for the problem. So once we have everything implemented, we were happy with that. We say, okay, so let's try now. Let's try to validate the model. Let's try to evaluate the predictive capability. So we made use of this experimental data that I mentioned before from Sanger et al., where we have kind of evolution of this migration dynamics on top of homogeneous substrate. Okay, so no such stiffness gradient at this stage. So at this stage, we did no fitting. So basically, we took some parameters directly from the physical meaning and from some experimental evidences. And what we can observe first in this panel A is that in black dots, we have the evolution of the experimental tendencies. And in red, we have the evolution of the model prediction. And this is basically distance to midline along time. So how this monolayer is expanding along time. So we can see a very good agreement by means of these migration dynamics between experiments and model. But we say, okay, so we're happy with that, but we need some extra validation, some double checks. So what is happening by means of mechanics? And then we compare experimental results with some models by means of stress traction. So we can see a very good agreement. Naturally, if we come here to panel B, we can see that the maximum stress traction from an experiment is almost identical to the one predicted to the model. So at this stage, we were happy with that. And we moved to test the model in heterogeneous substrate. So at this stage, we split all these simulations into main sets. So this first one, all these red lines are related to some stiffness dependent polarization. So we're going to say here, okay, so let's consider this propulsion force is somehow constant. And then everything is coming from this cellular polarization following this kind of gradient stiffness. And then in the other way around, so for the blue terms, we have that we skip this bit and we just focus on a propulsion force depending on the stiffness. So what we can see here is that by means of migration dynamics, we have almost same tendencies. So we can say that at this point, both hypotheses can explain this kind of migration system. But then we look at the, the result by means of mechanics. So let me go to the following slide, but this is, I think, more clear here. So what we can see is that this stiffness dependent polarization, we can predict this kind of migration toward the stiffer region. And if you realize we're keeping the symmetric distribution of the stress, and then we're just introducing kind of a slight increase in stress. But if we go for this kind of stiffness dependent propulsion force, what it implies is that we are losing this symmetry, and also it means that we're going to get kind of much higher internal stress. So I just wanted to highlight the usefulness of this kind of modeling approaches to complement experiments and to test hypotheses, right? Because at some point we have the experiments with some observation, we launch some testing hypotheses, but we don't really know which are the consequences by means of, for example, mechanics in this case. So at this point, let me jump to what I call private story and brain mechanics. So just want to make highlight a couple of points here. So just going to present some previous works I did actually during my postdoc at Oxford with Professor Antoine Jerusalem. So basically here, we were focused on central nervous system injuries. So that, okay, we have impact on our head. during sport-related activities, fall of a person, or even in a military context, we know there are some consequences by means of pathology, brain dysfunction, or even at the cellular level, we have some alteration in the action potential propagation, right? So at this point, we know there's some biomarkers such as sedative stress or others, but we thought, okay, so of course, one cause of that is that we have kind of mechanical deformation of the brain tissue itself. So there should be some mechanical variables governing all these CNS injuries. So in the end, we wanted kind of threshold saying, okay, so at this threshold, we can expect some injuries, below that not. So we were doing some collaborative work with people from Purdue University, and we realized that from an experimental point of view, it's almost impossible to track the whole event, looking what is happening inside. So we decided to go for computational modeling. And what I want to highlight here is that once we have some simple models, like the one I presented before, once we're happy with that validation and calibration, we can jump and go a step beyond, right? And we can now incorporate these models. Obviously, we will need extra validations for that, but we can incorporate that in more complex geometries, in more complex loading and boundary scenarios. And in that case, for example, we were able to see that in a closed-gate impact, we can have this kind of distribution of the stress and deformation within the brain, while in blast explosion, we can follow the whole mechanical event and see that the patterns of these mechanical distribution is completely different. So just wanted to close. It's not just conclusions, I would like to mention some ongoing work and future work. So basically, my conclusion here is that obviously mechanical environment, it's playing a major role. So my point now is, okay, let's try to control the cellular mechanical environment. Let's try to control this remotely. So what I want to propose here is to use some magnetoactive polymers, magnetoactive hydrogels really, to try to control the different mechanical properties or even mechanical deformations by the application of an external field. So the point here is that this is quite complex problem. And of course, we will need experiments, theory, and modeling. So yeah, this is what I'm really focused now. Some magnetoactive hydrogels, where we're going to have some mechanics that are interplaying with different magnetic fields, right? So applied magnetic field can introduce some mechanical changes. But of course, as we have this hydrogel nature, this is not a viscoelasticity just. So we will have also some solvent diffusion, some poroelasticity. So my idea here is, okay, let's try to build up kind of consistent computational framework so we can understand the physics, we can design properly this kind of substrate, and let's try to control the cellular mechanical environment. So I think I ran out of time, so don't know if there is question. If not, I'm very happy to have some contacts, look for collaboration. And just to mention, this is what this ERC starting grant is about. And well, happy to get feedback, comments, whatever you consider. Thank you very much.
Thank you, Daniel. Thank you for the nice talk presenting your research. So yes, we have 1 or 2 minutes if there is a question now. Otherwise, we will have time, as I said, at 1:20.
Nice.
So, well, I think it's good to be on time, you know? So we can continue. Thank you again, Daniel.
Thank you very much.